All right, so our next speaker is Christopher Ojukwu from University of Colorado in Boulder.
Thank you. Hello, everyone. My name is Christopher. I'm a PhD student at the University of Colorado, Boulder. And today I'll be talking to you about STIX, a method for detecting somatic structural variation and identifying novel fusions. Cancer caused by somatic structural variation, of course, is as a result of a change in the structure of the DNA. And just to remind us, this structural variance can come in different forms, like the deletion where a segment of the DNA disappears, or the duplication where a segment of the DNA repeats itself, or the insertion where one segment of DNA inserts itself into another segment of DNA. These structural variants have great implications in diseases, including cancer, and we see one of its effects in one of the ways
I think they're changing it manually.
Yeah.
So, does it work now?
Yes.
Okay.
Okay. Sorry for the small detour. So the structural variants have great implications in many diseases, including cancer, and we can see one of the ways in which it performs in, say we have a structural event like deletion. It rearranges the structure of the DNA, which means that two genes can come in close proximity to each other, and when this happens, a fusion occurs. And when I use the word fusion, I use that word very loosely, because in this talk, I could mean it as when a structural variant begins in one gene and ends in another, and this may or may not lead to a merge. So in this example, we see the common one seen in prostate cancer samples, the ERG-Tempest2. And a question that really encompasses our research is, how can the somatic structural variants be found? A way to do this, to look at it with single nucleotide variants, is by really just comparing a sample and the aligned reads to a reference genome. And by so doing, we can find any discrepancies between the sample and the reference genome. But of course, this method begins to fall apart with structural variants, because with structural variants, you have to take certain indications into consideration. Indications like the read depth and the discordant reads, because this raises certain problems like mapping errors and fragmented variants. And then the reference genome becomes inefficient in finding these. Another method for doing this is by using the tumor normal calling, which is where you really have a tumor sample and a normal sample, and you check to see if there is a variant in the tumor sample that also exists in the normal sample. But the problem with this also is a case of false positives, because all the attention is given to the tumor samples and not enough to the normal samples, which means that when an analysis is done, the normal samples have a case of high false negatives, and which in turn would affect the tumor samples, which in turn results to a case of high false positives. Our solution to this is really to use a population scale data set. And by doing so, not only are we able to find variants, we're able to discover how these variants affect diseases. And this is where STIX comes in, Structural Variant Index. With STIX, we're able to reduce the problem regarding structural variant detection into really a query problem. So how does STIX work? We begin by taking the aligned reads of a population. Of course, this aligned read would have both the discordant reads and the normal reads. And the normal reads are reads that have lengths as expected, and the discordant reads are reads that have lengths different from what we expect. We begin by taking away all the normal reads, and we're left with the discordant reads. And really from here, we index and it becomes a query problem. So for a given variant, say given a deletion, we find all alignments that supports this variant. And for this same sample and for this same variant, any other query would really be a noise. So with sticks, we have been able to filter SV calls and improve specificity. But what we're looking in improving is really sensitivity. And we do that by indexing and combing through disease-specific cohorts and healthy cohorts in a way of finding evidences that supports a fusion. So using my example, the ERK Tempest 2, say we have this variant, observe this variant in an individual, and then we turn this variant into the breakpoints of this variant into query regions. Then for a given analysis, we're really finding everything in a sample that supports this variant. All the evidence is in a population that supports this variant. But the problem with this is that the query regions are so restricted that it becomes really difficult to find enough samples. So I'll just show you some of our analysis. So we did this analysis and were able to discover in prostate cancer samples 6 out of 383. But we know that this number would be slightly wrong because the ERK Tempest 2 is pretty common in prostate cancer samples. So our solution to this is to use something called functional equivalent query, where instead of querying just certain query regions, breakpoints, we query entire gene bodies to find any evidences that supports this variant. So we've built upon this by indexing the PCAWG cancer samples, focusing on prostate cancer, and then the 1,000 genome samples, and then we count the number of evidences that support a fusion. But because we cannot rely on SV calls to give us a list of potential fusions, we take the count of all possible gene pairs, which is really approximately 20,000 by 20,000, giving us a resulting matrix. And from this point, we make certain assumptions. One of the assumptions we make with the 1,000 genome result is that we assume that it describes a neutral mutations and noise, which means by taking that away or eliminating that from the PCA result, we're able to arrive at novel fusions. So this is an example of our results. So this is for the cosmic fusions, and we see 2 outliers here, the ERK Tempest 2 and the GOPC ROS1. And of course, we expect that the depth of the ERK Tempest 2, the PCA depth will be high because we expect that we'll see that there. But we also see that it's relatively low in the 1,000 genome. But then again, we see the GOPC ROS1 that has a high depth in the PCA prostate cancer, but also a high depth in the 1,000 genome. But then what I'd like to draw your attention to is really the gene burden. So the burden is the frequency of evidences that connect one gene to another. So we see that example in the ERK Tempest 2, where we see that the gene burden is very low, but we see that the gene burden for the GOPC ROS1 is pretty high. And this tells us that this is a clear case of a false positive. So for that reason, we make ERK Tempest 2 upper bound, which means that at any given point, we're interested in everything that lies between the low left quadrant of ERK Tempest 2. So we see another example of this with the PCAWG prostate cancer mantle tumor normal fusions. We see where the ERK Tempest 2 lies, but also we see everything that falls in the lower left quadrant. And we're familiar with some of these genes because they're and some of these fusions because they've been well studied in cancer. But also we see some of the false positives in the top right that some of us might also be familiar with. But just the case, like we're saying, is the idea that with a high depth of the PCAWG, the prostate cancer, and a low depth of the 1,000 genome, and really a low gene burden, then we begin to know where or where not something might work. So this is a visualization of the 20,000 by 20,000 genes. So we see where ERK Tempest 2 falls. When we expand on that, we see that much very clearly. But then again, we see something that if we're saying that it falls in a high depth, but also a low 1,000 depth, we see that the reason why we're calling this a false positive also is because it has a high gene burden. And these are some of the fusions that we have confidence in. because they do show a high prostate cancer depth, a low 1,000 genome depth, but also they do show a low gene burden. But also, these fusions have been well studied, and it lets us know that the pattern we're tracing and the pattern we're creating, that we are on the right track. So with sticks, trying to reiterate, with sticks, there is no SV calling. You're really indexing and you're querying. And for the sake of increasing sensitivity, we're combing through disease-specific cohort and healthy cohorts to find novel fusions. I'd like to thank some of our collaborators from the University of Colorado Boulder and BioFrontiers Murad and Dr. Ryan Layer. I'd also like to thank our collaborators from the Baylor College of Medicine, Jincheng Zheng and Fritz Sedlacek. And Zheng actually has a poster this evening, number 178, if you'd also like to know more about sticks and the work we're doing. And thank you.
Thank you, Christopher. Questions? I'll maybe start with one. So so you focused on comparing prostate cancer to 1,000 genomes.
Yeah.
And Pan Cancer Project sequence many different tumor types.
Yes.
Have you thought about integrating all the different cancer types to look
yes.
at driver or fusions that may be, low frequency in one cancer type, but more prevalent? So if you look at everything?
Yes, we're currently working on that. We're actually looking at all cancer types available on PCAWG. Okay. And then we're sort of like building off of that. But we're starting with prostate cancer because we wanted to sort of like, and then with ERK Tempest 2, sort of like to have a benchmark of if we're familiar with this, then we can infer on other things. So yes.
Great, thank you.
Thank you.
From the floor.
So I noticed in one of the false positives that you showed, they were, just looking at the read name, they were annotated fusions, or like read-through transcripts. Did you, were there still structural variants that supported those?
Sorry, can you repeat the question?
So in the on the diagonal where you showed the false positives,
yes,
you can see it in the gene names themselves.
Yeah.
That they are annotated as
yes.
read-throughs.
Yes.
And when you study fusions, you get to know these.
Yes. Sorry.
And exclude them. But I'm wondering because they're recorded as read-through genes or fusion genes.
Mm-hmm.
Did you observe, do they have structural variants in them as well?
Yes, we take that into consideration when we're doing analysis, and sort of like, so when they have sort of like a high depth, we also have to take into consideration that some of them are read-throughs and some of them are sort of like embedded genes as well. So yes, we do take that into consideration.
Maybe just to follow on that question, like in PCOG, again, there's RNA-seq data for a lot of the samples as well. Have you thought about making an index over the RNA sequencing reads, and including DNA to maybe improve your power even more to see how things are expressed?
That would literally be my work through this break.
Okay,
So.
That sounds like a good break then. Any other questions from the floor then? All right, in that case, let's thank Christopher again.
Thank you.